Okay, it's 11:59 right now, and I have two very quick announcements. The first of all is for the graduate students, there are still openings to have a free lunch with Dr. Rieseberg after the seminar. And to everyone who is here and out in Zoom land, if you're in town, there's an open invitation at 5:15 today to have an informal social at Eastern Divide Brewery with Professor Rieseberg as well. So please feel free to invite yourself to that. It's now 12 o'clock, so I will start with the introductions. Loren, his academic pedigree is something I think is quite interesting for many of us. He received his bachelor's at Southern College in Chattanooga, Tennessee, and then went on to do his master's at the University of Tennessee in Knoxville and his PhD at Washington State University in Pullman. He then was an assistant professor at the Claremont Graduate School in Southern California, and then rose through the ranks of associate and full professor at Indiana University. He is currently endowed professor, the University Killam, sure I said that right, professor at the University of British Columbia in Vancouver. He has a really long list of awards, and you probably lose more time in talks about people going through the list of all of your honors and awards than anyone. But I'll just focus on a few of them to kind of give you a sense. He received the Darwin Wallace Medal from the Linnean Society of London. He is a fellow in many societies, including the Royal Society, which is the United Kingdom, part of the United Kingdom Academy of Sciences. He's a fellow of the American Academy of Arts and Sciences, American Association for Advancement of Science, yada, yada. The one that I think by far is the most coveted of all of his many distinguished awards was in 2003, he was awarded a MacArthur Fellowship. For those of you who are not familiar with MacArthur Fellowships, the foundation selects a number of people across all kinds of disciplines. And it's seeking individuals who have shown, quote, extraordinary originality and dedication in their creative pursuits. You probably heard of this on the news because each year when these come out, they're referred to not as the MacArthur Fellows, they're referred to colloquially as the MacArthur Genius Awards. And just to give you some idea of kind of the span of the types of people that earn these awards, in this past year, the past cohort of MacArthur Fellows ranged from a master hula dancer in San Francisco for maintaining and advancing that art form, all the way to Professor Lindsay Marr here at Virginia Tech for her contributions to understanding how SARS-CoV was transmitted as an virus. Really diverse group. Loren focuses his research typically on very deep biological questions. He then adroitly uses creativity to cut through complexity, resulting in always very original and impactful findings, such as those that he will tell us about now. Please join me in welcoming Loren.
Thanks, Don, for that very nice introduction. Somebody mentioned, though, that he came out and did a sabbatical with me in this 2 years, a year and a half or 2 years ago, in the summer and fall of 2022. And so it was a real pleasure to have him in the lab, and I got introduced to poison ivy. Well, I was introduced a lot when I lived in Indiana. Every spring I was put on steroids. That's probably why I got the MacArthur Award, was that creativity from all those steroids. I love those steroids, but apparently you're not supposed to be on them forever. In in British Columbia, we don't have them, so there's no excuse for that. Anyways, John introduced me to poison ivy, and again, I guess, to the genomics of poison ivy, and so it was a lot of fun to develop some basic genomic resources. Well, John developed them in the lab and created some genomes for poison ivy and poison oak, and now we're sort of, I guess, at the point of annotating the genomes, and he still has a number of others in the way. So that was a lot of fun. It was good to get to know him, and so I was really eager to come visit Virginia Tech. I do have colleagues here. Jason Holliday is someone who was at UBC, and I got to know him quite well. He was a grad student there. Anyways, what I'm going to be talking about today is sort of a very old problem in evolutionary biology, and that is sort of resolving the antagonism between divergent natural selection and recombination. And the basic idea is that divergent natural selection pushes or drives populations apart, and gene flow and recombination homogenize them, bring them back together. And this has been considered to be the greatest obstacle to speciation in sexual organisms. And the problem is quite straightforward, right? Let me see if I can move this green bar. No. Okay. Well, doesn't really matter. And I've illustrated the problem here in a very simple way, right? You can see a population of yellow daisies and a population of red flowered daisies. And if those populations to come into contact and all else being equal, sort of in a very simplest situation, they'll hybridize, exchange genes, and in just a few generations, they'll look the same. And so it's a fairly simple problem, right? And the early critics of Darwin's theory of natural selection were quite aware of it. They used different language, but basically they were asking how the changes brought about by natural selection will become established in a population when that population was still mating with maladapted individuals. In other words, it really was a gene flow recombination issue, even though they didn't know about recombination. And they also came up with a really simple solution, and it was a good solution, and that is geographic isolation. Obviously, if the populations are isolated by a geographic barrier, gene flow and recombination are not possible, problem solved. And in fact, certain very prominent, in fact, most evolutionists of the, some of them, of the 20th century, Ernst Mayr and others, went so far as to say that geographic isolation was required for speciation, that every speciation event had geographic isolation. The problem that came up is that, in fact, speciation with gene flow is common. And so my talk will really be really asking a very simple question, sort of is, how does this happen? And there's an emerging solution, and that is recombination modifiers. And the most famous of these are inversions and other kinds of chromosomal rearrangements. And basically, these suppress recombination in heterozygotes. You can see this here. Essentially, with many inversions, the recombinant gametes actually are inviable because they contain deletions and duplications, and so they're simply eliminated from the population, leaving you back with the non-recombinant parental allelic arrangements. And this allows them to keep sets of linked alleles together. It works very well if you just get selection against heterozygotes of those rearrangements. But you can continue to have recombination within in homozygotes for the arrangements. And so it reduces the sort of maladaptive effects of limiting recombination. And so here are some of the key papers sort of in the history of this idea. I should point out that this has sort of been an accumulating idea over the past 2.5 decades. And the first paper here, which I think was a really important paper, was a very simple simulation study by Adam Trickett and Roger Button. It's hardly been cited a lot at all in the literature. But they basically made a very simple observation, and that is that if you have suppression of recombination between a locus or a gene that's under divergent selection, and one that causes assortative mating, that will, of course, facilitate speciation. You don't even need a simulation to show that, right? You can just do it with a thought experiment. You keep these assortative mating and divergently selected loci together, and it's going to facilitate speciation. And however, as I mentioned, this was really sort of ignored in the literature. And then in 2001, there were two papers, one by myself. I hate to bring it up, but it was sort of I think, an important paper where I wrote a review about chromosomal rearrangements and speciation. And this was guided mainly by my own observations in sunflower, but I was aware of the Butland paper. And I basically made the argument that we were looking at the wrong thing. That most of the studies about chromosomal rearrangements had focused on their effects on fertility. And I said, we need to think about this. We need to start thinking about their effects. And then in the same year, Mohammed Noor, who's now at Duke University, also had a really influential paper where he showed that inversions could actually maintain genic incompatibilities in the presence of gene flow. And both of these arguments focused on the role of these rearrangements in limiting gene flow through their effects on recombination. But neither Mohammed or I solved what was really an important question, and that is actually one of the reviewers asked me about it in this paper. And that is, well, how are these inversions becoming, or other rearrangements becoming established? And this really wasn't solved until a paper in 2006 by Mark Kirkpatrick and Nick Martin, where they showed that the suppression of recombination between locally adapted alleles, that is alleles that are adapted to the same environment, actually will facilitate the establishment of inversion as long as this happens in gene flow. You have to have gene flow for this to work. So what I'm going to do now is to actually sort of talk about how we've accumulated some data that sort of provides some of the missing pieces in this story, looking at wild sunflowers. And the first part of the talk will be just looking at the genetic basis of adaptation in wild sunflowers. Remember, local adaptation is key to the establishment of these inversions and other recombination modifiers. I'm then going to talk about a couple of case studies of speciation with gene flow. One in the very earliest stages of speciation between two ecotypes, flowering time ecotypes of the silverleaf sunflower. And then the second, we'll look at a late stage of speciation with gene flow involving dune versus non-dune ecotypes of the prairie sunflower. And then I'll end the talk by addressing this issue of how do sunflowers avoid the maladaptive effects of recombination suppression? So let me introduce you to the wild sunflowers. They are native here to North America. There are about 50 species. And for our purposes, what's really interesting is that they often have populations that are tightly locally adapted to different environments. And we typically call these ecotypes. And this local adaptation is important because at least in sunflowers, but also in many other group of plants, this local adaptation represents the first step in speciation. And I also pointed out that the cultivated sunflower is derived from this wild common sunflower that is found throughout much of the United States and plays a large role in this particular story. And so we started out, so this is first looking at the genomics of local adaptation. And this study started out with a 50,000 kilometer collection trip by a really excellent botanist in my lab, Dylan Burge, was a postdoc in the lab. And Dylan had a red truck, which he just drove all around North America and Mexico. And the reason that we wanted him to take the truck is because we also collected soil from each site and 4 soil samples from each site. And so he collected a couple tons of soil during this collection trip. And you'll notice that he went all the way up into Canada and down into Mexico. However, most of my later slides do not involve the Mexican populations because he got to the border coming back from Mexico. He had all his permits in place, and the lovely US Border Service destroyed all of his collections. So next summer he redid this, and this time he just shipped the samples directly to Canada. But by that time were already running the common garden experiments, and so they were not included in the analyses. But I now have a grudge against the US Border Service. All right, so what we first did was ran some common garden experiments with all of these collections. Here at UBC, this is the Helianthus annuus common garden. And this work was spearheaded by Natalia Berkovich, who John knows well. Natalia was the one who, she's a research associate in my lab, and she trained John in the various methods for DNA isolation and sequencing and so forth, and library preparations. And then we did, we phenotyped them for about 80 or so phenotypic traits, as well as we also determined the soil characteristics of each of the collection sites, and also downloaded 25 climate variables from the World Wide Web, World Climate site. And this work was partly done also by Marco Tedesco, who also John met, who is now an assistant prof at UBC. And then we resequenced them. And this is just shows the, and I want to point out Greg Owens and J.S. Lagar were postdocs who did the bioinformatics. And essentially, here's the sort of phylogeny based on the resequencing data. And you can see the cultivated sunflower coming out within wild annuus. And then this is Helianthus argyphilus, the sister species that I'll be talking about some. And then these are the prairie sunflowers here. You can see they're a bit more distantly related to the other two that I'll be discussing. And we ended up with anywhere between 70 and 200 and so million single nucleotide polymorphisms, or SNPs. And so we chose really high quality sets of 4 million each for each of the species for the analyses that I'm going to show you. And then we did something that I think most of you are familiar with. It's something called genome-wide association mapping. And so all this means is that you look for associations or correlations between genotypic variation, in this case, these single nucleotide polymorphisms, and phenotypes that we collected from individuals that were collected in the natural population, in this case, grown in the common garden experiments to reduce environmental variation. And then in this case, I'm showing you one of the traits that we looked at. These are ultraviolet nectar guides. And this is what sunflowers obviously look like under visible light, but under ultraviolet light, you can see that they have these sort of bullseyes or UV nectar guides that vary in size. And this is for Helianthus annuus, this is for Pedalaris. And then you can look for associations. So this is a Manhattan plot. Each of these here on the bottom is a chromosome, and then the significance threshold is on the y-axis, and you can see the Bonferroni corrected threshold is in red, and you can see that this is a particularly unappetizing GWAS screen. There's nothing that looks particularly promising there. You might have sort of a bit of a peak here, but it's nothing very convincing, and not something I would spend time sort of searching on the genetic basis of. On the other hand, at Helianthus annuus, we found that what we were really looking for, and this is a nice peak here on at the bottom of chromosome 15. And if you look under that peak, it actually maps to the promoter region of a sunflower homologue of Arabidopsis MIB111. And it turns out that MIB111 regulates the production of flavanols, which are known to absorb UV light. And in fact, Marco did further work to show that, to functionally functionally confirm that this indeed is the gene that's responsible for the variation we see in ultraviolet nectar guides. But the other approach that we took for looking at local adaptation was something called genotype environment, GEA analyses. And in this case, instead of using sort of morphological phenotypes, here we used characteristics of the environment as the phenotype. And so you're doing the same thing, right? You're looking for associations between SNPs and features of the environment. So this is a GEA analysis of continentality, which just means these are environments that have more extreme temperatures. But you see the same basic pattern for different temperature-related environmental factors. And in this case, one of the best hits was to this gene. This is a homologue of Arabidopsis heat intensity 1, which has been shown to be related to heat tolerance in Arabidopsis. And in fact, this has already been used now by sunflower breeders to enhance heat tolerance of sunflowers. All right, so far those analyses were exactly what we predicted. We had thought we'd be able to find, by doing these analyses, these GWA and GEA analyses, we'd find genes that were of interest to us that were involved in local adaptation. But the surprise came was that we found large signal plateaus for some traits. And that is, in this case, we would see large groups of genes that actually were being inherited together, were acting together on a particular trait. And in the literature, these are sometimes called supergenes. These are cases where you have, or haploblocks or supergenes. These are where you have cassettes of genes that are acting evolutionarily as a unit. That is, they affect the block of traits are inherited together, and they have block-like effects on phenotypic traits. And these are actually quite large. Just to give you an idea, the largest one is here. So this one is about 30 megabases. This one is about 60 megabases. This one here is the largest one, Helianthus annuus. It's 101 megabases. To give you some idea, that's about 2/3 of the Arabidopsis genome. All in this one supergene. It contains about 1,600 genes. And one of the questions we wanted to know is, are there more supergenes? And so for this, one of my grad students, or postdocs at the time, Greg Owens, used an approach called local PCA population structure. It's called LOSTRUC. But the basic idea is it's a windowed-based analysis where you look for the regions of the genome that are acting differently from other regions of the genome. In this case, you make a PCA of population structure. So each of these windows, we run a PCA on each window across the genome. And generally, you can see that they show the same population structure, the same relationships among populations, until you get about 150 megabases down the chromosome, and all of a sudden, they're very different. And so these turn out to be a really good way of finding supergenes. These are regions of the genome that show a very different population structure, and thus are probably under selection than other parts of the genome. And as you might guess from my earlier comments, most of these supergenes turn out to be inversions. This is just information from reference sequences, but initially we used evidence from like genetic maps and from high-C sequencing and so forth. But the most convincing evidence obviously comes from comparing reference sequences. And what you see here, here's a small inversion. This is about 4 megabases on chromosome 1, and this is that one I just shown you on chromosome 5. It's about 30 megabases. This is the 101 megabase inversion on chromosome 13, and then there's a smaller one. And you can see these here. So sometimes we find inversions, small inversions within larger inversions. In this case, we do have that, but we also have what I think is a misassembly. And so, and we found that most of these supergenes turn out to be very ancient. This is just an example from chromosome 5 I showed you previously. This is the phylogeny of this supergene. And here is haplotype 0. It's found where it should be, as sister to Helianthus argyrophilus. But here you find that haplotype 1 is actually older, originated prior to the diversification of all the annual sunflowers. It's about 2.5 million years old. And if you look at all of these, you find that In fact, in most cases, they either predate the split of the subspecies or the species. There are a few younger. And we find that these supergenes underlie traits associated with local adaptation. This is just showing sort of for different trait categories, plant architecture, carbon nitrogen content, development, inflorescence, seed, leaves and seeds, and these are climate factors and soil. And you can see that we have that in many of these, most of the supergenes actually are associated with these various features. That is, they control local adaptation to many of these different features of the environment. And this is just a summary. In this particular study, we identified 37 of these supergenes. And the size ranges from about 1 to 100 megabases. Most are inversions, they're old, and they underlie many phenotypic traits and environmental variables. So what I want to do now, though, is to say, okay, we know a little bit about the genetics of local adaptation. We know that these supergenes are important. Now let's sort of relate this to the process of speciation. And as I mentioned, we're going to be talking about these ecotypes in sunflower. And they were discovered, this is the silverleaf sunflower. It's found on the Gulf Coast of Texas. And they were discovered by one of my former grad students, Brooke Myers, and she discovered that there was a short early flowering form that's found mainly on the barrier islands. And then there's a taller late flowering form that's found inland. And this makes sense. The climate in the barrier islands is much milder than the one inland. And so typically the inland plants they actually are woody annuals of all things. They basically sit there in the summer and do nothing when it's really hot. And then when it gets cooler in the fall, they flower and produce seeds. Whereas the early flowering ones just flower throughout the entire season. And Brooke did some work, did some reciprocal transplants in Texas, and she found that indeed that the early flowering was indeed favored on the barrier islands, whereas late flowering was favored inland. And these are not the cleanest data that I would like, but that was because there was a drought that year and a lot of the plants died, and so the signal's not quite as strong as we would have liked. But the genetics are very clear. So we found that there was an enormous supergene on here at the end of chromosome 6. It's about 30 megabases. And you can see that this is a huge signal. It's, I think, the largest signal I've ever seen in at least one of our own GWAS experiments. And it explains about 80% of the flowering time variation between these two ecotypes of Helianthus argyphilus. And one thing I want to show you is there's some interesting features, right, of this region. You can see that there's kind of a gap here. We'll talk about that gap. There's also a really strong signal this part, and then it gets weaker towards the end. And if you look at this more closely, you find that this actually is very well correlated with recombination. It turns out there's no recombination at all in a 10 megabase region here in the middle. And then we have a handful of recombinations for the end, and that has reduced the strength of the signal. This gappy area here in the middle contains a gene family that's quite famous. This is a gene family. This is the flowering locus T, which is it's the protein of flowering locus T that travels from leaves to the apical meristem in the shoot to trigger flowering, and sometimes called the hormone florigen. And we had previously discovered this cluster based on just genetic mapping data a number of years ago, and we had found that one of these genes, HAFT1, was involved in during domestication, resulted in a change in flowering time. It was a frame shift mutation in FT, which resulted in the flowering time shift. So we were interested that, again, action was coming to this particular cluster of genes, and FT in particular seemed to be involved. It was missing in all of the late flowering plants, present in the early flowering plants, and of course heterozygotes, and present also in the heterozygotes, but in lower frequency. And so Marco went in and showed that indeed FT1 is not amplified from genomic DNA in the late flowering plants. It's also not expressed in the late flowering plants. And he was able to functionally complement the Arabidopsis knockout, indicating that the FT1 from early flowering Argophilus is indeed functional. So in this case, we were able to figure out exactly where this supergene came from. It comes from introgression from Helianthus annuus. So this is a phylogeny of the supergene. The annuus is in, although all the populations of annuus are in the orange, the populations of Argophilus are in green. So the late flowering Argophilus supergene is here in its right position as the sister, and here's the early Argophilus. So this was derived from introgression. So it provides an interesting example where a gene was lost in a species and then regained through introgression, and has important functional effects in adapting Helianthus argophilus to the barrier islands. So what about speciation? I've talked mainly about adaptation, and local adaptation, of course, is a step in the progress of speciation. So what about assortative mating? What about reproductive isolation? One would think that this would have a big effect on reproductive isolation. Well, not as much as we had predicted. And that's partly because there's environmental variation in Texas. What we find is that on the coast, we get fairly strong assortative mating in November, where there's mostly only the late flowering, ecotype flowering. We see the opposite thing on the island, where we get fairly strong assortative mating in July, where almost the only ecotype that's flowering is the early flowering ecotype. In the center there, we get very weak reproductive isolation. So it's the kind of thing, whereas there is partial reproductive isolation. By the way, this was done by a grad student, Marianne Pasquale. She basically took all of the seeds from these different populations where you had both ecotypes, genotyped them, and found out exactly who was mating with who. So it was a lot of work. So obviously this is not as strong a barrier as we had initially anticipated. So we also, though, look to see, well, how much is this supergene contributing to differentiation between these two ecotypes? And that's shown here. So what we're showing here, these are all chromosomes, and now we're showing FST. FST is a common measure of differentiation. And what you can see is that in this chromosome 6, the supergene does show fairly high differentiation between the two, between the island and the mainland, but it's not unusually high. Here is another inversion that I haven't talked about on chromosome 10 that also plays a role in this early differentiation between these ecotypes. But you can see this is really the very earliest stages of speciation. Whether these things go on to form another species really is unclear at this point. Okay, so now I want to move to a second case where we're much further along in speciation. In fact, some might consider these to be good species, and this involves the dune and non-dune ecotypes of the prairie sunflower. And these are, by the way, the mega sand dunes in the Great Sand Dune National Park and Preserve in Colorado. And you can see these yellow patches. Well, these are the dune sunflower ecotype. And then we get the non-dune ecotype, yes, off the dunes on the sand sheet below. And the biggest difference between them is seed size. The dunes have very large seeds. The non-dunes have small seeds. And you can see some obvious reasons for this, right? Larger seeds will reduce the chances of burial by sand, as well as provide the resources to go deep into the sand dune to obtain water and other nutrients. So this is the geographic distribution of the prairie sunflower. It's found throughout much of the central Great Plains, And in a couple places, it has colonized these sand dunes, once in Colorado and once in Texas. It also has colonized some Canadian sand dunes that we're beginning to look at. So this is the situation in the Great Sand Dune National Park. These are these 200 to 230 meter mega dunes. They're the largest dunes in North America. And then down here below, you have the sand sheet. And then behind you have these mountains. And the dune plants, they have several characteristic adaptations, and these also are maintained in the greenhouse. They flower later, they have larger seeds, and they have greater nitrogen use efficiency. The dunes, as you might guess, are very low in nutrients. And the first thing we wanted to establish, and this was work done by Rose Andrew, a former postdoc, was to see whether there was ongoing gene flow between the dune and non-dune populations. And what we found was that indeed there was gene flow. It was somewhat asymmetric. There was between about 2 and 6 immigrants per generation from the dune to the sand sheet, and about 0 to 0.6 to 4 the other direction. And we used coalescence models for this, and we're able to show that the ecotypes diverged less than 10,000 years ago. This makes sense. There was during the last ice age, it was too cold to have sunflowers on the dunes. And we found that gene flow was moderate, but high enough to prevent differentiation through drift alone. And so this does look like a situation of very recent speciation in the presence of gene flow. And one of my other students, Kate Ostovic, was just a remarkable field biologist. She did reciprocal transplants on the dune with both hybrid and parental individuals. And you can see this is a challenging habitat. I destroyed so many careers with these dunes. People would go down there and their experiments would be destroyed by blowing winds. You come back the next day and there was no experiment. Also, you notice the sand dunes, there's not a lot of green stuff there. You transplant out some nice green sunflowers, and next day, you think there are no herbivores out there. There are lots of herbivores out there, and they're just gone. So Kate managed, I'm not quite sure how. So this is the dune garden, that's the sand sheet garden. And she found pretty much what we expected. She found that indeed large seeds were favored in the dune habitat. And in the sand sheet, it really didn't make any difference. Both large and small seeds did just fine. This shows the seed size in blue of the non-dune seeds, and then here the dune seeds, and then the F1s are in purple. You can see a large maternal effect, and then by the back process, they're kind of homogenized. And so the dune seeds, right, are favored in the dune habitat, but they make no difference, and they do fine in the non-dune. But in the non-dune habitat, the non-dune plants are taller, produce more seeds, produce more flowers and produce more seeds. So it's sort of a classic trade-off. And if you use Astor models that combine fitness across the life history, you get the classic sort of pattern of local adaptation where non-dune plants are favored in non-dune habitats, dune plants in dune habitats, and then hybrids do poorly in both. So there is a strong fitness cost to being a hybrid in the system. And Kate also went through and calculated the strength of reproductive isolation across the life cycle. This is something that it's sort of become a cottage industry in studies of speciation. People look at reproductive barrier strength across the life cycle of plants. And so in this case, she's showing that immigrant and viability is very strong. That is, if you take a dune seed and put it in non-dune and vice versa, you get this strong immigrant and viability. There's a weak assortative mating due to flowering time. There's a larger difference due to pollinator isolation. This is because there are different pollinators on the dune versus the non-dune system. And in fact, Kate found 2 bees that had never been seen before or never been new to science. We found a weak sort of conspecific pollen precedence barrier. What this means is that dune moms preferred pollen from dune dads, and non-dune moms preferred pollen from non-dune dads. We can't figure out yet how they figured that out, how they recognize them, but somehow that happens. But we see no loss of fertility or inviability or fertility. We do see strong selection against hybrids. And so this results in a total reproductive barrier strength of between 0.993 to 0.996, so almost complete isolation. And you say, well, how do we see evidence of migration or gene flow between them? As long as the effective population size is high, you can still get a trickle of gene flow, even if you have a very strong reproductive barrier. So that's how you sort of reconcile that apparent discrepancy. So I haven't talked about the supergenes, and you know, we had hints of these things in Rose's study and so forth, but we didn't actually know for sure that these were indeed supergenes. But once we analyzed these in detail, we kept on finding them showing up. You can see here, this is a region, that chromosome inversion on chromosome 5 I've shown you before. It pops up here on the dunes in Texas and Colorado and Texas. This one on 9, Colorado, Texas. You can see the pattern repeating here, is that these inversions or supergenes tended to be found on the dune and non-dune systems. And we find when we do the GWAS and GEA analyses that indeed the supergenes control dune adaptation. For example, here we're showing seed size, 3 of the supergenes, days to flowering. Again, you can see a supergene effect. And cation exchange capacity, this is a measure of soil fertility. And so you're really looking at tolerance or resistance to low nutrient soils. And again, you can see that this maps to these 3 supergenes. And if we look at the reproductive barriers, we can also infer which of these reproductive barriers are housed or associated with the inversions. And we find indeed that immigrant inviability, flowering time, pollinator isolation indirectly, all, and extrinsic ecological selection against hybrids all map to the supergenes. We've been struggling to map this particular trait, the conspecific pollen precedence, so we don't know about that. But in general, you can see that, in fact, the reproductive barriers do indeed map to the supergenes. Another sort of independent way of looking at the role of the supergenes in adaptation and speciation is to look at selection against heterozygotes. And in this case, we're looking in the dune population. We find that, and here we're looking at the change in frequency between seeds and flowering or mature plants. And you can see that all of the heterozygotes have reduced in frequency, except for this one on chromosome 14, which I can't figure out. I think it's just a sampling error issue. So you can see that they're all selected against, which suggests that they indeed are selected against in the dune environment. Okay, so just sort of we already talked about how these dune-specific traits map to the large supergenes, but now we can also look at genetic differentiation, because this gives you an idea of how important they are to speciation. And here we find, here's FST, and you can see that most of the significant differentiation, or most of the differences between the dune and non-dune ecotypes, are defined actually by the supergenes. So this probably provides the strongest evidence of their role in driving the origin of these dune ecotypes in the presence of gene flow. Okay, so another mystery then is, how did these ecotypes form so rapidly? I mean, 5,000 years is what we're probably looking at, which is very, very fast for a speciation event. And what we think is that probably these ancient supergenes were likely recruited from standing variation. And I've illustrated that in a very crude diagram here. So this is sort of a standing variation in the non-dune ecotype. And the idea is that they were then recruited into the dune ecotype. So the idea is that these dune ecotypes have probably originated again and again and again. In other words, speciation didn't happen just once, didn't happen just twice. It probably has happened many, many times during the history of sunflowers. And they just form again and again after changes during the glacial cycles. Okay, so now I want to switch gears and talk about this other issue. So we've sort of established that these play an important role in speciation, but if they're so important in speciation, then why do we have recombination at all? And we know that recombination is valuable. And I've sort of illustrated the reason why we care about recombination is that it provides a way of reducing, increasing the efficiency of both purifying and positive selection. And I've illustrated this here. So typically on a chromosome, right, you're going to have both deleterious and beneficial mutations. I'm not a great artist, so I use smiley faces for this. But the idea here is, so here you have chromosomes that have both deleterious and beneficial mutations. By crossing over, you can bring the deleterious mutations together, the beneficial mutations together. And so this allows selection to eliminate the chromosomes carrying the deleterious mutations and to bring the beneficial mutations to higher frequency. And so in general, recombination is considered to be beneficial in most circumstances. Yet we have all of these supergenes, various inversions and other chromosomal structural variants that are quite beneficial in leading to speciation in sunflowers. So how is this happening? How are sunflowers avoiding these maladaptive effects of recombination suppression? And when we looked at this, so now it's quite, you can take a look at any given chromosome or genome and find out, look at the accumulation of deleterious mutations. There are a number of different ways you can do this. One favorite way that's pretty clear is you look at the fraction of nonsense mutations. These are the ones that change an amino acid to a stop codon, and so you don't really want them. And here we're comparing the fraction of nonsense mutations genome-wide versus in the inversions. And some of these inversions are very large. And so in order to do this fairly, what we have to do is, because recombination has a really large impact on the accumulation of deleterious mutations, we have to fractionate the genome into different recombination categories. So this is the part of the inversion where you have no recombination or basically none in homozygous individuals. Here you have reduced recombination, here high, but you see the same pattern in all of them, right? There's no difference between the genome-wide accumulation and the inversion. The same thing is seen here for this chromosome inversion on Helianthus argyphilus, and the same thing here on this inversion in P-Paris. This one actually is significant, but in this case, there's actually less load in the inversion than in the non-inverted region. So what's going on? Well, one thing you always have to remember is that one of the key aspects of inversions and other chromosomal rearrangements is that in heterozygotes, it's only in heterozygotes that you get recombination suppressed. In homozygotes, you can continue to have recombination. So it turns out that in the sunflowers, because these inversions tend to be geographically segregated, they're more homozygous than expected by chance, which enhances recombination. So this is shown here using U-scores, essentially, and what you can see is that the inversions are much more homozygous than the rest of the genome on average, which allows, and because recombination can take place in these homozygous orientations, then this reduces the impact or the accumulation of deleterious mutations. Okay, so just to wrap up, I've shown that these sunflowers are differentiated by massive supergenes. Well, many of them are massive. They contribute importantly to local adaptation and reproductive isolation. They're generally caused by these large inversions or other structural variants. We haven't shown all of the kinds of rearrangements that cause this recombination suppression. and they act as recombination suppressors. And the point I want to make is they act as ideal recombination modifiers because recombination is suppressed in heterozygotes only. So this typically will allow full recombination to continue in populations, but will not allow recombination between populations. So that's ideal for driving divergence and leading to speciation. They play a major role in evolution by reducing recombination between locally adapted alleles, as well as, or in addition to those, contributing to reproductive isolation. And they're often ancient, and so it looks like they contribute to repeated speciation in time and in place. And the one thing I haven't mentioned is that these are super common in other groups of plants and animals. Now that we have genomic data, they're just popping up everywhere. I think I saw, I mean, I keep on getting these papers to edit or review, and I think I saw one, keep on seeing the numbers just keep going up, but like Mango has 16, and you know, it just is just they're phenomenally active, phenomenally frequent, and in my view, they offer a general solution to how speciation can occur in the presence of gene flow and recombination. So basically solving that old mystery that was first sort of suggested by the critics of Darwin's origin of species by natural selection. And I want to thank all the people who did this work, because as John knows, he almost never sees me in the lab. I'm not sure if I ever saw, I'm not sure he ever saw me in the lab. But these are the people, Marco, Tedesco, Natalia, Greg, that I've already mentioned, also Kate, Deas, Kai Chi, they're all that played a really important role in this. This is an old picture of my lab, but I imagine that you probably recognize most of them, John. And then I also have collaborators, particularly at the University of Georgia, and funders. I put cannabis up there because the Canadian government has become increasingly reluctant to fund sunflowers, and so the cannabis industry has been indirectly funding the sunflower operation. So thank you.
Lots of time for questions.
I was wondering if you look into the origin of these versions, the context that elements that you know, but they have top.
You know, we have looked some at this. It's something that we haven't fully finished yet because the reference genomes sometimes don't cover these regions as well as we would like. But there is certainly an association between these regions with high levels of LTR retrotransposons and inversion breakpoints. Of course, these things are the dominant fraction of the genome. And so it looks like, you know, when we run the tests, it looks like it seems to be significant, but, you know, I'm still sort of a little bit leery that we fully. Yeah, so that's the best explanation we have at this point in terms of the breakpoints.
See a similar thing in plant cells.
In what?
Plant cells, they actually, the fruit fly study has a really high proportion of LTRs as well. It's funny.
Oh, it's in land snails, because I've seen the marine snail stuff, but not the land snail stuff. That's, yes, they always, but land snails, do they move very much? I mean, how much gene flow is there?
Very little. I mean, like, it depends on the species, but microtons.
You know, yeah, well, yeah, okay. That's my only. You do see, you know, not in all, there aren't always involved, inversions aren't always involved in speciation with gene flow. Sometimes they're under balancing selection within populations, so involved in seasonal changes in weather like in Drosophila. So I'd be interested in knowing what they're doing.
So I think one of these 100 plots, would the sample size play a role in affecting the plot to actually detect anything in the other species, because I could see it's the difference in the sample size is fivefold.
Yeah, so the point here is that we had difference in power in the different experiments. So in Annulus, you had the highest number of populations, whereas in Pedalaris, we thought we were going to have the highest number, but it turned out that there were 3 fairly differentiated groups within what we thought was Pedalaris, and so the sample sizes were larger, and so you get lower power to detect supergenes or QTLs or so forth. And so that is a factor. In this case, it's really weird for us. We were able to detect a phenotype or environment associated with all of the supergenes in almost all of them in Pedalaris and Argophilus. But in Annulus, we actually have some we haven't been able to nail down, which had the largest sample size. So you know, it must affect power, but probably more in terms of effect sizes rather than maybe significance, in the sense that we had more success in, well, we had probably more success in identifying individual genes in Annulus with the larger sample size, but for the Heppleblots, the sample size didn't help us for some reason. We're actually using other methods to try to figure out what's going on. I mean, you can't have 101 megabase region and have it affect nothing. And so we don't know if there's just recombination within it or what's going on, but we're sort of using some other methods to figure it out. But yeah, there was large variation.
So I was thinking about the hypothesis about that 5,000 years. So you have the existing emergence and somehow selected to a new environment. And I wonder how frequent that inversion has to be for this to occur, because you have this selection against type, right? If you have, I would imagine that those inversion will be relatively low in frequency to begin with. But how is that maintained? And then when you have a new environment, you can quickly adapt.
This is a real mystery because we did not find those inversions at, you know, very high frequency outside of the dunes, for example. And so, yes, it is something that we're assuming that they're being maintained, but you know, that it's possible there are some, if you look throughout it's possible, for example, that there might be low dunes where they're favored or something like that. But certainly on the sand sheet below, you would think that they would be selected quickly to very low frequency or simply eliminated from the population. So I think that is sort of we're not sure whether we have these sort of, whether they're just at low frequency in the standing variation or whether they're sort of almost, what would you call it, in reserves or sort of refugia where they're being maintained at higher frequency. There are places like, for example, in Utah, where you do have fairly extensive dune systems. These species don't typically occur there on them. There are other species that occur there. So I'm guessing that there must be some low dunes or swale habitats that these things might, that we haven't discovered, that these things would hang out during the Wisconsin glaciation. At this point, we really need to go find those populations and show that the inversions actually exist there. There was one hint where I don't know if you remember those figures I showed where showing the inversions were at the dunes. There was one population way out in Arizona where it had one of the inversions. So yeah, but we haven't found any populations that are sort of off dunes that they're at high frequency, or even typically we don't find them at all.
Just as far as sunflower biology, are they all self-compatible?
These are all self-compatible outcrossers, yeah. So, which does change the dynamics a lot.
Any more questions? Please join me in thanking Loren.
In the graduate students who are interested in having a free lunch,